Imagine your cells had a master switch, a molecule so fundamental to life that without it, your cells cannot produce energy, repair damaged DNA, or regulate the genes that control how fast you age. Now imagine that this molecule declines by up to 50% between your 20s and your 50s. And imagine that scientists at Harvard, MIT, and the Buck Institute for Research on Aging have been quietly racing to understand how to restore it. That molecule is NAD+. And the compound that is making headlines across the longevity world right now as the most promising way to boost it is called NMN. Whether you have heard about it from a podcast, seen it trending on YouTube, or noticed it in a supplement store, NMN is not just another wellness fad. It is one of the most seriously researched longevity molecules in the world today, and what the science is revealing about its potential to slow, and in some cases reverse, key mechanisms of biological aging is genuinely remarkable. Right now, researchers from some of the most prestigious institutions on Earth are publishing findings on NAD+ and NMN at a rate that is accelerating every year. The U.S. Senate has spotlighted NAD+ precursors as one of 6 key areas for future longevity research. Celebrities from Joe Rogan to Jennifer Aniston have shared their personal experiences with NMN supplementation, and longevity experts like Dr. Mark Hyman and Dave Asprey have called NAD+ one of the most important molecules for controlling the aging process. But beyond the celebrity endorsements and the hype, what does the actual science say? Today, we are going to break it all down. We will cover what NAD+ is, why it declines, what NMN does inside your body, what the clinical evidence actually shows, and most importantly, what you need to know before spending a single dollar on a supplement. Welcome to the channel. If you are new here, this is your home for honest, science-backed longevity and biohacking content. No hype, no pseudoscience, just real information you can use to live better and longer. If that is what you are looking for, hit subscribe right now. Tap that notification bell, and share this video with someone in your life who is serious about their health. What we cover today could change how they think about aging at the cellular level. Let us get into it. To understand NMN, we need to first understand NAD+, because NMN is simply the most direct pathway your body has to make more of it. NAD+ stands for nicotinamide adenine dinucleotide. It is a coenzyme found in every single living cell. Without NAD+, your cells cannot perform 3 of their most critical functions. First, energy production. NAD+ is essential for converting the food you eat into ATP, the energy currency your cells run on. Without adequate NAD+, your mitochondria cannot generate energy efficiently. Second, DNA repair. Your DNA is constantly being damaged by environmental stressors, oxidative molecules, and errors in cell division. NAD+ fuels the enzymes that scan for and fix this damage. Without it, errors accumulate, and cellular aging accelerates. Third, gene expression regulation. A family of proteins called sirtuins, often described as longevity genes, require NAD+ to function. Sirtuins regulate inflammation, stress response, metabolism, and the rate at which your cells age. The problem is this: NAD+ levels decline significantly and consistently with age. Research has shown that by the time most people reach their 50s, their cellular NAD+ levels can be 50% lower than they were in their 20s. This decline is not a minor inconvenience. It is a fundamental biological shift that affects every cell in your body simultaneously. Less NAD+ means less efficient energy production. It means slower DNA repair. It means sirtuins that cannot do their job. And it means a body that is aging faster at the cellular level than it needs to. This is the problem that NMN is designed to address directly. NMN stands for nicotinamide mononucleotide. It is a naturally occurring molecule found in small amounts in certain foods, including broccoli, avocados, edamame, and cabbage. However, the amounts found in food are extremely small. you would need to eat several kilograms of broccoli daily to approach the doses used in clinical research. This is why supplementation has become the primary focus of researchers and health enthusiasts interested in raising NAD+ levels effectively. Here is how NMN works at the cellular level. When you take NMN, it enters your cells and is rapidly converted into NAD+ through a series of enzymatic steps. The key enzyme in this process is NAMPT, nicotinamide phosphoribosyltransferase, which is the rate-limiting step in the primary NAD+ biosynthesis pathway. NMN essentially provides direct substrate for this pathway, bypassing some of the earlier steps that can become bottlenecks with age. The result is a measurable and relatively rapid increase in intracellular NAD+ levels. Multiple human studies have now confirmed that oral NMN supplementation raises NAD+ in blood and tissues within weeks of starting supplementation. It is worth briefly addressing the comparison between NMN and another popular NAD+ precursor called NR, or nicotinamide riboside. Both NMN and NR are converted into NAD+ inside cells, and both have research support. NMN is one biochemical step closer to NAD+ in the biosynthesis pathway than NR is. Some researchers suggest this makes NMN more directly efficient for raising NAD+ levels. However, the honest picture is that both compounds have shown efficacy in clinical studies, and the debate between them is not fully settled. What matters most for longevity purposes is the downstream increase in NAD+ activity, which both compounds appear capable of producing. Once NAD+ levels are raised through NMN supplementation, the downstream effects involve several key biological systems. Sirtuins, which are among the most studied longevity regulators in biology, become more active. SIRT1, for example, regulates gene expression in ways that reduce inflammation, improve insulin sensitivity, and activate pathways associated with caloric restriction, one of the most well-documented longevity interventions in biology. SIRT3 regulates mitochondrial function and protects against oxidative stress. SIRT6 is critically involved in DNA repair and genomic stability, and its activity has been directly linked to biological aging rate in multiple species. PARP enzymes, which are responsible for detecting and repairing DNA strand breaks, also require NAD+ as a substrate. When NAD+ levels are low, DNA repair activity slows. Errors accumulate in the genome over time. This genomic instability is one of the hallmarks of aging identified in the landmark 2013 paper by David Sinclair and others that defined the 9 hallmarks of aging. By raising NAD+ and restoring PARP activity, NMN supplementation may directly support the genome maintenance systems that keep aging cells functioning properly. The CD38 problem is also worth understanding. CD38 is an enzyme that consumes NAD+ and becomes increasingly active with age and with chronic inflammation. It is sometimes called the NAD+ drain. Research has shown that as we age, CD38 activity rises sharply, which is one of the primary reasons NAD+ levels fall even in people who are otherwise healthy. This is why some advanced NAD+ formulas are now combining NMN with compounds like apigenin or resveratrol, which have been shown to inhibit CD38 activity and reduce NAD+ breakdown. Raising production while reducing consumption is a dual strategy approach that makes biological sense. Let us move to the evidence, because this is the part where longevity supplements most often disappoint. The mechanism looks compelling in the lab. But does it translate to real human outcomes? With NMN, the answer is more encouraging than with most longevity compounds, though important nuance is required. The foundational human safety and pharmacokinetic trial was conducted by researchers at Keio University School of Medicine in Japan and published in the journal Cell Metabolism. This was the first randomized, double-blind, placebo-controlled trial of oral NMN in healthy adults. Participants received 500 milligrams of NMN daily for 10 weeks. The results showed that NMN was safely metabolized without causing any serious adverse effects. Blood markers for liver function, kidney function, and hormone levels all remained normal. NAD+ metabolites in blood increased significantly in the NMN group. This trial established the foundation for human NMN research and confirmed that oral NMN does reach circulation and raise NAD+-related metabolites. A clinical trial published in Science, one of the most prestigious scientific journals in the world, examined NMN supplementation in postmenopausal women with prediabetes. Participants received 250 milligrams of NMN per day for 10 weeks. The results showed that NMN improved muscle insulin sensitivity, specifically the ability of skeletal muscle to take up glucose in response to insulin. This is a critically important outcome for longevity, because insulin resistance in muscle tissue is one of the primary drivers of metabolic aging, type 2 diabetes, and accelerated biological aging. The improvements in insulin signaling were accompanied by increased expression of genes involved in muscle remodeling and energy metabolism. This was a landmark result because it demonstrated a real functional human outcome, not just a change in a blood marker. A study published in NPJ Aging and Mechanisms of Disease examined the effects of NMN supplementation on physical performance and muscle strength in older men. Participants over 65 received 250 milligrams of NMN or placebo daily for 12 weeks. Those receiving NMN showed improvements in grip strength and walking speed compared to the placebo group. Walking speed and grip strength are validated markers of biological aging and are among the strongest predictors of health outcomes and mortality in older adults. Improvements in these markers from a supplement intervention in a 12-week randomized trial represent a meaningful and clinically relevant finding. Research has also examined NMN's effects on sleep quality, which is deeply connected to both NAD+ metabolism and biological aging. A Japanese clinical trial found that NMN supplementation improved subjective sleep quality and reduced daytime drowsiness in older adults. Sleep is when NAD+ dependent DNA repair and cellular maintenance are most active. The fact that restoring NAD+ may improve sleep quality creates a potential positive feedback loop where better sleep further supports NAD+ dependent repair processes. What about the bigger picture? Animal studies, which are further along in the research timeline than human trials, have consistently shown extraordinary results with NMN. In mice, NMN supplementation has improved energy metabolism, reversed age-related decline in physical performance, restored fertility in aged female mice, improved cardiovascular function, and extended health span. These animal results are the reason leading researchers like Dr. David Sinclair at Harvard have been so vocal about the potential of NAD+ restoration. However, it is important to be honest. Mouse results do not always translate to humans, and the human clinical data, while promising, is still relatively early stage. Long-term safety and efficacy data in large human populations is still accumulating. Expectations should be calibrated accordingly. Understanding why NAD+ decline matters so deeply requires zooming out to look at the broader biology of aging. In 2013, David Sinclair and colleagues published a landmark paper identifying 9 hallmarks of aging, which are the core biological processes that drive aging across species. NAD+ decline and its downstream consequences directly intersect with at least 5 of these 9 hallmarks. That intersection is why longevity scientists consider NAD+ restoration to be one of the most upstream and broadly impactful interventions in the aging field. The first hallmark is genomic instability, the accumulation of DNA damage over time. As we discussed, NAD+ fuels PARP enzymes that repair DNA. When NAD+ falls, DNA repair slows and genomic instability accelerates. The second hallmark is epigenetic alterations, changes in how genes are expressed that do not involve changes to the underlying DNA sequence. Sirtuins, which are fully dependent on NAD+, are primary regulators of epigenetic marks across the genome. When NAD+ falls, sirtuin activity drops, epigenetic regulation becomes dysregulated, and genes that should be silenced become active, while genes that should be active become silenced. This epigenetic drift is one of the clearest molecular signatures of biological aging. The third hallmark is mitochondrial dysfunction. NAD+ is the primary electron carrier in the mitochondrial electron transport chain. Without adequate NAD+, Mitochondria cannot generate ATP efficiently. They produce more reactive oxygen species, which damage surrounding cellular structures. And they are more likely to become the dysfunctional, energy-depleting organelles that accumulate with age and drive the fatigue, cognitive decline, and metabolic deterioration that many people attribute to simply getting older. The fourth hallmark is cellular senescence, where cells stop dividing but refuse to die and instead secrete inflammatory molecules that damage surrounding tissue. Research has shown that NAD+ depletion accelerates the accumulation of senescent cells, while NAD+ restoration can support the pathways that clear them. The fifth hallmark is altered intercellular communication, particularly the chronic low-grade inflammation that researchers call inflammaging. Sirtuin activity, which requires NAD+, directly suppresses inflammatory gene expression through regulation of NF-κB, one of the master switches of inflammatory signaling. When sirtuins are inactive due to NAD+ deficiency, inflammatory tone rises. And chronic inflammation is one of the most powerful known drivers of accelerated biological aging across virtually every tissue in the body. This convergence across multiple hallmarks of aging is what makes NAD+ restoration so theoretically compelling as a longevity intervention. Most supplements and interventions target one pathway or one tissue. NAD+ sits upstream of 5 foundational aging mechanisms simultaneously. Restoring it is not a single target intervention. It is an attempt to restore a core biological resource that the entire cellular machinery depends on. One more area worth highlighting is the connection between NAD+ and the circadian clock. Research has shown that NAD+ levels fluctuate rhythmically across the 24-hour day, synchronized with the circadian clock. Disruption of circadian rhythms accelerates NAD+ decline and vice versa. This means that lifestyle factors like consistent sleep timing, regular meal timing, and morning light exposure are not just circadian health strategies. They are also NAD+ optimization strategies. Conversely, NMN supplementation may support circadian rhythm stability, particularly in older adults whose clock mechanisms have become less robust. This bidirectional relationship between NAD+ and circadian biology adds another layer to why restoring NAD+ levels matters for the full picture of biological aging. The science is compelling. Now let us talk practically. What do you actually need to know if you are considering NMN supplementation? We will cover dosing, timing, quality selection, cost, and the lifestyle factors that matter most for NAD+ optimization. On dosing, the human clinical trials to date have used doses ranging from 250 mg to 1,000 mg per day. The majority of positive findings in human studies have come from the 250 to 500 mg range. However, many longevity researchers and practitioners who use NMN personally, including Dr. David Sinclair, report taking higher doses in the 500 to 1,000 mg range. The honest answer is that the optimal dose for longevity outcomes in humans has not been definitively established. A reasonable starting point for most people is 300 to 500 mg daily. You can assess your response over 6 to 8 weeks and adjust as needed. Starting at the lower end is prudent, particularly if you are new to NAD+ precursor supplementation. On timing, most researchers recommend taking NMN in the morning. This aligns with the natural circadian rhythm of NAD+ biosynthesis, which peaks in the morning and early afternoon. Taking NMN in the evening may not align optimally with the body's natural NAD+ production cycle, and there is some concern that elevating sirtuin activity late at night may interfere with sleep in certain individuals, though this is not well established. Morning dosing with or after breakfast is the standard recommendation. On supplement quality, this is where careful attention is essential. The NMN supplement market has grown rapidly, and product quality varies enormously. Look for supplements that provide third-party testing certificates of analysis confirming purity and potency. Look for manufacturing in GMP-certified facilities. Look for a stated purity percentage for the NMN ingredient itself, ideally above 96%. Be cautious of products that make extraordinary longevity claims beyond what the science supports. And be aware that some products labeled as NMN have been found to contain significantly less of the active compound than the label claims. Third-party verification is not optional. It is essential. On cost, NMN supplementation at effective doses runs approximately $30 to $80 per month, depending on the brand, dose, and delivery format. Liposomal formulas, which claim enhanced absorption through encapsulation in lipid particles, tend to cost more. The bioavailability advantage of liposomal NMN over standard capsules is real in principle, but has not been definitively proven to translate to meaningfully better NAD+ outcomes in published human trials yet. Standard high-quality capsule forms are well absorbed and have the most human research behind them. If budget is a primary concern, a reputable capsule form at 300 to 500 milligrams is a reasonable choice over a more expensive liposomal product with less established clinical data. Now the critical lifestyle context. NMN supplementation is not a substitute for the lifestyle factors that are the most powerful drivers of NAD+ levels. Exercise is the single most potent known stimulus for NAD+ production. High-intensity interval training and resistance training both increase NAMPET expression, the rate-limiting enzyme in NAD+ biosynthesis, in muscle tissue. A study published in Cell Metabolism found that exercise raised NAD+ levels in muscle more powerfully than NMN supplementation alone. Caloric restriction and intermittent fasting activate AMPK, a metabolic sensor that upregulates NAD+ producing pathways. Quality sleep, as we discussed, supports the circadian regulation of NAD+ biosynthesis. And reducing chronic alcohol consumption is important, because alcohol metabolism consumes NAD+ at an extraordinary rate, depleting cellular stores rapidly. There are also honest limitations to name. While the human clinical evidence for NMN is more robust than for most longevity supplements, it is still early. Most human trials have been short in duration, typically 8 to 16 weeks. Long-term safety data extending beyond 1 year in humans is limited. Some researchers have raised questions about whether chronically raising NAD+ might have unintended consequences in certain contexts, including theoretical concerns about supporting certain cancer cell populations, though no evidence of this has emerged in human trials to date. As with all supplements, consulting with your physician is important, particularly if you are managing a chronic health condition or taking medications. Your practical action plan is this. Start with lifestyle first. Commit to consistent exercise, prioritizing both resistance training and aerobic work. Practice intermittent fasting or time-restricted eating if appropriate for your health status. Protect your sleep quality and consistency. Reduce alcohol consumption. These interventions raise NAD+ through your body's own biosynthetic machinery, which is always the most sustainable and side-effect-free approach. If you then want to explore NMN supplementation, choose a third-party tested product from a reputable brand. Start at 300 to 500 milligrams in the morning. Be consistent for at least 8 weeks before evaluating effects, and consider testing your NAD+ levels with a blood test before and after to assess your individual response. Track objective metrics like HRV, sleep quality, and energy levels alongside any blood testing. The bottom line is this. NAD+ is not hype. It is a fundamental molecule in the biology of aging, backed by decades of serious research and an accelerating body of human clinical evidence. NMN is the most direct and well-studied way to restore it. The science is promising, the safety profile is reassuring in the short term, and the biological logic is sound. For those serious about longevity, maintaining NAD+ levels as you age is a strategy worth understanding deeply and approaching thoughtfully. The research is still maturing, but the signal is clear enough that some of the world's leading longevity scientists are not waiting. And now, neither should you wait to understand what the evidence actually says. Thank you so much for watching all the way through today. If this video gave you real value, please like it and share it with someone who cares about their health. Subscribe if you have not already, and drop a comment below telling me whether you have tried NMN or NAD+ supplementation, and what your experience has been. I read every single comment, and I genuinely want to hear from you. See you in the next one.